This session is about proteins and amino acids. We'll understand what amino acids are, what proteins are. We'll try to understand the different chemical properties of amino acids, the different types of amino acids. We'll also try to understand the different types of proteins, the different levels at which proteins are understood structurally, etc., etc. Now let us just start with what proteins are. Now, proteins are the building blocks of life. This is what you might have studied in your junior classes also. Proteins make up nearly all the structural components of your body. For example, nails, they are made up of keratin. It's a structural component. The hair, right? The horn of the rhino is made up of keratin. Our muscles, our tissues, our connective tissues, they are rich in a protein called collagen. Right? Similarly, there are a wide variety of other structural proteins that build the structure. Certainly, there are inorganic minerals such as calcium and phosphorus, which also contribute to the buildup of structure in living organisms. In certain cases, they make up bones, right? In our cases. Apart from that, what is the other function of protein? Proteins are also an essential component which drive all metabolic reactions. Now, what do we mean by metabolic reaction? The chemical reactions of life, all of them are called metabolic reactions. For example, respiration itself, the breakdown of glucose or any other respiratory substrate. For example, even fats can be broken down for respiration. So the breakdown of molecules is metabolism. Also the building up of molecules, for example, proteins or fats, synthesis of carbohydrates, or building up of DNA. All of these are metabolic reactions. Under normal circumstances, in the absence of proteins called enzymes, these reactions will proceed extremely slowly, very, very slowly. So enzymes are absolutely essential in increasing the speed of these reactions, and also these reactions, the specificity of this reaction is, or the preciseness, the precision of these reactions is enhanced by the presence of enzymes. So, two functions of proteins: they build up the body and they drive metabolic reactions. They increase the speed and efficiency of chemical reactions, all chemical reactions of life. But what are proteins? Should be our next question. And a protein, out here in this particular image, we have a strand of protein. We can consider it a protein. Now, a protein is a polymer. Now, what is a polymer? A polymer is is any particular compound. It can be any particular chemical species which is made up of repeating units, repeating nearly similar units. And in this particular case, the protein is made up of repeated units which are called amino acids. So certainly amino acids differ from each other in certain ways, but they are amino acids. So the different amino acids, when they are connected to each other, you see they are building up this long chain-like structure. So what is this? Protein is a polymer made up of monomeric units or single units called amino acids. So each of this bead is an amino acid as shown here. So these are all amino acids, and amino acids are connected to each other by a bond called the peptide bond, which we'll see in some time. And also, okay, let us have a look at the diagram. A particular protein has something called as the N terminus and C terminus. N simply means the end with a free nitrogen, and C terminus means the terminus with a free carbon. So these are the two ends for further bonding if necessary, if required. Proceeding further. Now let us try to understand the properties of amino acids, but before we look at the properties, here is the image of an amino acid. Now, amino acid, let us look at the first point, can be considered as a substituted methane. I hope you people know the structure of methane. However, if you do not, even if you do, let me just draw methane for you. So, methane has one carbon atom in the center, and as it is a tetravalent molecule, it has to bind with 4. And what are these 4 atoms that bind to the central carbon? They are hydrogens. So this is a methane molecule. Now, why do we call protein? Why can we consider an amino acid? My apologies. Why do we consider an amino acid a substituted methane? Because each hydrogen, barring one. So let us say this is the carbon. This is the central carbon. The one of the hydrogen atom remains unaltered, right? But if you look at this particular hydrogen, it is substituted by a functional group called the carboxyl group, COOH, carboxyl group. It's an acidic group. This particular group provides acidic character to the amino acid, acidic. If you look at let us look on the opposite side first. If you look at this particular hydrogen, it is substituted, replaced by another functional group which is called amine, NH2. Not 3, not 4, NH2. And there is no charge on this, there is no charge on this at the moment. Right? And let us now look at this, the last hydrogen atom which is substituted. This last hydrogen atom is substituted by something called as the R side chain. The letter R, you can have any letter out here, you can have X, Y, Z, anything out here. The letter R signifies that this is a variable side chain. R can be, it may be simply a hydrogen atom, it can simply be a CH3 group, or it can be a branched carbon chain attached to the alpha carbon. Now coming to alpha carbon, the central carbon, why is it called the alpha carbon? The central carbon is called the alpha carbon, and alpha carbon is basically the carbon to which functional groups are attached. So we see this carbon has a carboxyl group attached to it, an amine group attached to it, and so we call it a alpha carbon. Likewise, this carbon of the carboxyl group, or the entire carboxyl group which is attached to the alpha carbon, is called the alpha carboxyl group. The amine functional group which is attached to the alpha carbon is called the alpha amine group. Simple. Now, so we have basically discussed what an amino acid structure looks like, and you should know the structure, you should be able to draw this. Proceeding further, the next property of amino acid is amino acids when they are in this solution that have a neutral pH. Now, let us assume, let's say distilled water. If we take distilled water, what is the pH of distilled water? It is 7. The pH is 7, and pH 7 is neutral. It is equally acidic, equally basic. It is a neutral pH does not mean it is neither acidic nor basic. It actually means it is equally, the acid and base cancel out each other, and that's why the pH is neutral. So anyways, when an amino acid is placed in an environment which is neutral, the amino acid, any particular amino acid, occurs as a zwitterion. Zwitterion. That's a very pleasing term to hear, zwitterion. Anyways, so what is a zwitterion? Let's come to that. Now, zwitterion is any molecule. It can be any molecule. Or that molecule should suffice a condition, which is a molecule that has equal positive and negative charge on it. Let us have a look at this amino acid. So what do we have here? We have an alpha carbon, we have a R side chain, we have a hydrogen. So there are no charges on carbon, no charge on hydrogen atom, no charge on the side chain. But what do we have here? We have the NH2. Oh, this is 3, but I'm saying NH2. Let's go back to the basics. NH2, amine group. This is the general structure of an amino acid. So at neutral pH, the carboxylic group, we have COO negative, but see, what is the original structure of an amino acid? COOH. So when the pH is neutral, the COOH, the carboxylic group, gets deprotonated. And let's have a look here. At neutral pH, the carboxylic acid, the carboxylic functional group, gets deprotonated. It loses its proton, and where does that proton go? It gets added up on NH2. Let's go back to the image. So basically, this proton is lost and this goes here. So hydrogen ion, when it is added on to this amine group, it will have an extra positive charge. And as a hydrogen positive ion is lost, there is a slight negative charge on oxygen. So what do we get? At neutral pH, we get an amino acid which acts as a zwitterion. It has one positive charge, it has one negative charge. Right? Equal positive, equal negative charge. Any molecule that has equal positive and negative charge is called a zwitterion. So amino acids at neutral pH act as zwitterions. But what happens when an amino acid is placed or finds itself in an acidic environment? So at low pHs, when the pH is low, it means the environment is acidic, and when the pH is high, it means the environment is alkaline or basic. Now, when the pH is acidic, the carboxyl group, see what is happening to it. At neutral pH, it lost a proton, but at acidic pH, because there are so many hydrogen ions around, what is an acid? An acid is a proton donor. So acidic environments will be rich in hydrogen ions. So now there are so many hydrogen ions that the hydrogen gets added back See, the hydrogen gets added back here, carboxyl, and we get back a carboxylic group. So at acidic pHs, the amino acid gets protonated, specifically the carboxyl group gets protonated, and hence the entire amino acid has a net positive charge. It is no more a zwitterion, it has a net positive charge, right? Because now only the amino group has a positive charge, an extra proton is present. What happens to an amino acid when the environment or the surroundings are alkaline? Now, under alkaline conditions, the amino acid, it gets deprotonated, which means the amine group, you see, zwitterion condition, neutral pH. When you add an alkali to an amino acid, this particular amino group, I'm sorry, I've just botched it a little, so let's erase it. And so what do we see here? The charged amino group, it loses a proton in alkaline conditions. So a proton has been lost from here. As the positive charge has been lost from here, what happens to it? The entire amino acid now has a net negative charge. And why does it have a negative charge? Because the carboxyl ion is still negative. So under alkaline conditions, the amino acid acts as an anion, negatively charged ion. In acidic conditions, an amino acid acts as a cation. And at neutral pHs, any amino acid will act as a zwitterion, which is equal positive, equal negative charges. A very important property. Proceeding further. Now we'll move on to the next subheading, which is the types of amino acids. What are the different types of amino acids? Now, the property of any particular amino acid is determined by, number one, the functional groups and the side chain. This is the most important part, the side chain. See, because let us have a look at this particular image here. Now, this particular image contains the structures of the 20 major amino acids that are found in the living systems. So, if you look at all of these amino acids, you will see that certain parts are consistent. There is a carboxylic group, carboxylic group, carboxylic group. Have a look at the others, carboxylic group, carboxylic group, carboxylic group. Alpha carbon is in the center, alright? So all of them have an alpha carbon. You look at any of these amino acids, they have an alpha carbon. An alpha carbon has carboxylic group attached to it. Yeah, all of these amino acids will have an amino group attached to it, an amine group or amino group attached to it. But how do all of these amino acids differ from each other? They differ from each other in the R side chain. So look at the side chain here. It's simply a hydrogen atom in case of an amino acid called glycine. In case of alanine, it is a methyl group. In case of valine, it is a branched alkane, or basically a hydrocarbon. Here you can see even a more complex hydrocarbon chain. Out here, what do we see? We see rings. Right, ring structures. So all of the amino acids that differ from each other on the basis of the R side chain, and also the property of amino acids, whether the physical properties or the chemical properties, are determined, okay, by functional groups, but most importantly by the R side chain. Now, how can we classify amino acids? Because we are going to categorize amino acids. There are various ways of categorizing amino acids. The first way is on the basis of the structural nature of the side chain. How the side chain is. Is the side chain actually a chain, a branch chain, or a linear chain? Have a look. This is a linear chain. This is just a single molecule. Here we have got a branch chain, again a branch chain. Here we have got a slightly complicated chain with a structure. And in this particular case, let's have a look at this. This is not a chain, rather a ring. In this case also we have a ring. In this case we have a ring here, and we have a ring here also. So phenylalanine, tryptophan and histidine, these particular amino acids are called aromatic amino acids because they have a ring attached in there or present in their side chain. Whereas all the other amino acids are called aliphatic amino acids because they have a hydrocarbon chain attached to them, a long chain, not a ring. Right, so this was one of the ways of classifying amino acids. The other method of classifying amino acid is based on the chemical property of side chain. The previous one, the previous characteristic was the structural nature of side chain, but this time we are going to classify amino acids on the basis of chemical properties of side chain. What is the property of this side chain? What is the chemical property of this? Now, certainly all of these are different side chains, different types of side chains, so their chemical properties will also be different. Now, on the basis of the chemical properties of side chains, these are all side chains. Amino acids can be classified into 4 types, and they are polar amino acids, non-polar amino acids, acidic amino acids, and basic amino acids. Now, let us start with non-polar amino acids. Now, non-polar, I hope from your previous classes you know what a polar molecule is. A molecule that has a separation of positive and negative charges on it. A molecule. Let us take for example water, H2O. Now, hydrogen has slight positive charge, oxygen has a slight negative charge. What do we see here? That the charges are separated. So, negative charges on oxygen and positive charges on hydrogen atoms. So, such a molecule is called a polar molecule, where we see separation of charges on the molecule. A zwitterion is also a polar molecule. If you look at a zwitterion, it's also a polar molecule. It has a separation of charges. Anyways, proceeding further. So what is a non-polar amino acid? Let's start with this non-polar amino acid. A non-polar amino acid has a hydrocarbon-rich side chain. Hydrocarbon. What is a hydrocarbon? Hydrocarbon, as the name suggests, any molecule compound which is only made up of carbon and hydrogen together. Right? So let us look at some polar, non-polar amino acids. Let's have a look at this. So carbon, hydrogen side chain. There are only carbon and hydrogen. Let's have a look at this. Only carbon and hydrogen. Only carbon and hydrogen. Right? Let us also have a look at a couple of others. Let's have a look at this. Only carbon and hydrogen. Okay, certainly there is another group out here which complicates it. So let's come back to something simpler. And alright, so these were simpler. Right? Only carbon and hydrogen. So a non-polar amino acid has a side chain. Remember, all the side chains have been highlighted either in green or in probably pink and blues and maybe dark pink, right? Anyways, so non-polar amino acids have a hydrocarbon-rich side chain, and these amino acids, non-polar amino acids, are poorly soluble in water, right? Now, let us look at the next type of an amino acid, which are called polar amino acids. Polar amino acids do not have hydrocarbon-rich side chains. They have polar molecules attached to them, which means they have a certain charge on them. Let us have a look at some examples. So these are polar side chains. Have a look at this OH. Have a look at this OH here. So alcohol group is a polar group. Then we got sulfur-hydryl group, SH group. This is also polar. OH is a polar group. NH2 is a polar group. Similarly, NH2 out here is a polar group. So these are amines or alcohols or sulfur-hydryl groups. All of these are polar groups. They interact very intimately with water. They love water. And so all of these polar amino acids dissolve pretty well in water compared to non-polar amino acids. So this is the difference between non-polar and polar. Let's come down to the third type of an amino acid based on the chemical property of side chain. See, it's only on the basis of side chain that we are classifying amino acids. So acidic amino acids, when they are put in neutral solutions, right, the side chains act as acids. They donate protons. Slightly difficult. Let's try to understand this. So an acidic amino acid at neutral pH. So let's go back to this image. So let us assume for a moment that this is this side chain will not be hydrogen here. It will be something else. Let's get down here. So let's take an example of an acidic. So this is an acidic amino acid. Now, in this amino acid, we have the alpha carbon, we have got the carboxyl ion, we have got the amino group out here, which is in the ionic state. So this is basically the zwitterionic state. This is not the zwitterionic state. But at neutral pH, this particular amino acid has the side chain, the R side chain of this particular amino acid is behaving like an acid. So, aspartic acid, it's also called aspartate. Aspartic acid, I'm just going to draw the side chain of aspartic acid. It is CH2, it is C double bond O, OH. H is missing here, if you see. H is missing here. Why? because at neutral pH, this proton is donated, is given out. So this particular amino acid, aspartic acid, has lost its proton to the surrounding. Anything that loses its proton or gives away its proton is called an acid. So what is the summary of it? An acidic amino acid, when put in any environment which is neutral, will behave like an acid. It will donate protons. So the examples of acidic amino acids are aspartic acid and glutamic acid. Likewise, we have basic amino acids. When a basic amino acid is put in a neutral environment, how does the R side chain or the variable side chain behave? It gains a proton. Remember, bases are proton acceptors and acids are proton donors. They give away protons, bases take in protons. So the example of basic amino acids are lysine, arginine and histidine. Let's take a simple example here. So you see in this side chain we've got this NH3 plus, right? Let me just write it here. So we've got NH3 plus. Under non-neutral conditions, the amino acid lysine will have NH2 as the last part of this chain. But when this amino acid is put in a neutral solution, what it does, it takes up a hydrogen ion from the neutral surroundings. How does that happen is something that you will learn in the higher classes. So it takes up a proton and it gets a positive charge, right? It acts like a base, proton acceptor. So I hope you understood what are the different types of amino acids based on chemical properties. We've got non-polar amino acids, hydrocarbon rich, poorly soluble in water. We've got polar amino acids, which are excellent or wonderfully soluble in water, but they do not have polar side chains. We've got acidic amino acids, which at neutral pH act as acids, and we've got basic amino acids, which under neutral environmental conditions act like bases or proton acceptors. Proceeding further. Now, the next article is how are proteins built? Now, proteins are built by amino acids, and amino acids are linked to each other via a peptide bond. Let us try to understand how it works. So, what do we have here in this diagram? We've got two amino acids. We've got one here, we've got one here. So, this is in the center, we've got the alpha carbon. Here we've got the amine group. Here we've got the carboxyl group. The carboxyl group of this particular amino acid, the amine group of this particular amino acid, the side chains. Now, in the formation of a bond, a peptide bond or bonding between two amino acids, the carboxyl group of one amino acid and the amine group of the other amino acid are involved. So, what happens? It is a simple dehydration reaction. The two amino acids, they come close to each other, and there are these enzymes which are involved. Again, proteins that are involved in the synthesis of peptide bond. And in this particular peptide bond formation, the hydroxyl, the OH group of carboxylic acid combines with the hydrogen of the amine group of the other amino acid. They combine together, they form water. So such a reaction is also called a dehydration reaction. So this is a So this is called a dehydration reaction because water is removed. When the water is removed, you see this carbon and this nitrogen, they combine together. They are connected to each other via a bond, via an attractive force. And this bond here is called the peptide bond here. This is a peptide bond. So amino acids are linked to each other via a peptide bond. Now when two amino acids are linked to each other, they form a structure. So this structure here, two amino acids are linked to each other, they'll form a structure which is called a dipeptide. Similarly, if there are three amino acids attached to each other, it's a tripeptide. 4 is a tetrapeptide, and so on and so forth. But we don't use all of these terms. We simply use a term called a peptide. So a short chain of amino acid is called a peptide. A longer chain of amino acid, which is longer than 50 amino acids, is called a polypeptide. Different types of polypeptides or same kinds of polypeptides or even a single peptide can act as a protein. So what is a protein? So basically here we are looking at the structural hierarchy. Amino acids, they combine together to form a peptide. Peptides, longer peptides are called polypeptides, different polypeptides or similar polypeptides, or even a single polypeptide will make up a protein. So there is a structural complexity, structural hierarchy, and a structural difference between these different entities. Amino acid peptides are larger than amino acids, polypeptides are larger than peptides. Protein is the largest and the most complex of all of these combinations. Proceeding further. Now, how many amino acids are actually known to us? Well, there are over 100 amino acids known to us, but only 20 amino acids make up all proteins in all the forms of life present on our planet. And what are these 20 amino acids? They are given out here. Now, there are so many of them. Each amino acid can be represented by 3 letters. For example, glycine can be represented or written as GL pi. Similarly, proline can be written as PRO, tryptophan TRP. It is not necessary that only the first 3 letters are chosen. You will have to remember them, you will have to learn them. Methionine is MET, fortunately the first 3 letters. But if you look at other examples, for example, let us look at asparagine. It's not ASP, it is ASN. So, that is the first thing. Now, each protein, this is also a very important point, each protein is unique in the type of amino acid it has, in the number of amino acids it will have, and in the sequence of amino acid, which amino acid is connected first, which is second, which is third. Each protein differs from each other in the type of amino acids, in the number of amino acids, and in the sequence of amino acids. It is not necessary that a protein should contain all these 20 types of amino acids. It will contain a combination of them, but it is not necessary that all 20 may be present. They may or may not be present. Proceeding further. Now let us try to classify proteins. Now proteins can be classified in many ways, and the 2 major ways are, first of all, we can classify proteins on the basis of what function do they play in the body of an organism. So you got structural proteins and you got functional proteins. So out here I have drawn, this is a structural protein, keratin. It is present in nails and hair and hooves, etc., etc. And here I have got another protein called nuclease. These are the actual structures, the computer generated structures of proteins. Now what is a structural protein? A structural protein is characterized by it being rigid. Look at your nails, they are pretty rigid. Water insoluble. When you wash your hands, your nails do not get dissolved. Structural proteins are also stable, they are long, they are like strings. So these are the properties of structural proteins, and an example is keratin and collagen. So what do we have here in this image? We have a hair, a strand of a hair, and the finest structure, or this hair, is made up of these coiled string-like proteins, and these are keratin chains. These are those proteins. So the first type of proteins are structural proteins. They'll make up the structure of the body. Example, keratin, collagen. The second type of proteins are called functional proteins. Proteins that perform a certain function in metabolic reactions. So enzymes, all the enzymes are functional proteins. Here we have an enzyme called nuclease, and the function of this particular enzyme is to break down nucleic acids like DNA and RNA. Now, what are the characteristics of functional proteins? They have a 3-dimensional shape which can change. So the shape of this protein can change under different circumstances. For example, if there is a change in pH, change in temperature, change in moisture, the structure of this protein can change. Examples of functional proteins: hydrolases and polymerases. You will learn in the higher standards or in some future classes what these enzymes are all about. Now, proteins can also be classified on the basis of what shape they are of and what is their solubility. On the basis of shape and on the basis of solubility, proteins are of 3 types: the fibrous proteins, fiber-like proteins, the globular proteins like globules, and membranous proteins present inside the membranes. And here we have them. So this protein here, it is made up of fibers. You can see there are 3 fibers colored with 3 different colors. So this is a fibrous protein, and this is basically a collagen protein. Here we have a globular protein. You can see it is like a globule. It is spherical. And here we see a membranous protein, a protein which is found inside the membrane. Now the membrane can be a cell membrane, can be the membrane of the nucleus, can be the membrane of endoplasmic reticulum, or the Golgi, or the plastid, or some other organelle, or some other structure. So very rapidly, let's just go to what fibrous proteins are. They are simple proteins. They are regular and linear structure. Linear is straight line like structure. They are insoluble in water. These fibrous proteins are insoluble in water. Example is again collagen and keratin. You have to think of your nails. Globular proteins are usually spherical. Have a look at this. This is nearly like a sphere, right? They are usually spherical and they are water soluble. These proteins are water soluble. So most of the enzymes are globular proteins. Now membrane proteins, these are associated with cell membranes. So we have them here. Proceeding further, now we can describe the protein structures at 4 levels. Any particular protein, let's take an example, keratin. Let us take an example, keratin. So keratin protein or any other protein for that matter can be understood at 4 different levels, and these are called levels of organization. Now keratin protein can be understood as simply a chain of amino acids, simply a sequence of amino acid. And so this level of organization, where we study a protein only as a sequence of amino acid, is called the primary structure. So this is called the primary structure of proteins. This is the very basic level of protein structure. See, just the sequence of amino acid. When you are simply describing the sequence of amino acid in a protein, you are describing the primary structure of a protein. Now, all of these amino acids, if you remember, let's have a look at this particular image. The side chains have a negative charge, have a positive charge. Even under different pHs, they can act as zwitterions. These chains, these polypeptide chains or these proteins chains, the amino acids can interact with each other. So this particular amino acid, for example, can bond with the other one through a hydrogen bond, through a weak force. This particular amino acid can make a bond here. This particular amino acid can make up a bond here. And if such bonds are formed, what will happen to this linear chain? It will fold on itself. Now there are 2 types of foldings that are seen. The first folding is known as the alpha helix, and the other type of folding is called the beta pleated sheets. So these are alpha helix or beta pleated sheets. So any particular protein will fold either as an alpha helix or as a beta pleated sheet. And now this level of organization, the coiling of the linear protein molecule, when you are describing it, we call it the secondary structure of the protein. Right? Now let's come to the tertiary structure. 3, and there is a knot on the top. 3 knot. This tertiary, this numbering system basically means this word tertiary. Now in complex proteins, you will have a large number of proteins which are found as alpha helixes. You will find many proteins which will be found as beta pleated sheets. So in complex proteins are basically a combination of alpha helixes and beta pleated sheets. Let's look at this. So when the structure that arises due to the interaction between alpha helixes and beta pleated sheets, we get a higher structure called the tertiary structure. So this is the 3rd level of protein structure, which is called the tertiary structure. And eventually in extremely highly complex proteins, which are a combination of many secondary structures, many tertiary structures, we get a even more complex level of protein structure, which is called a quaternary protein structure. So basically quaternary protein structure is seen in those proteins which contain more than 1 amino acid side chains. So anyways, this is all about the different levels of protein structure. And with this we come to an end of our presentation on amino acids and proteins.